Hi, welcome to Fun Science with Rohini. After Boveri and Sutton proposed the chromosome theory of inheritance, and after proof for the chromosome theory of inheritance was provided by Thomas Hunt Morgan, further studies on DNA as the molecule that passes on the characters came from Griffith and Avery McLeod in McCarthy. Griffith performed his transformation experiment in 1928. Avery McLeod and McCarthy performed their experiment in 1944. But both are related because the conclusion that can be drawn from both these experiments is that DNA is the transforming principle. DNA is the one that passes the traits. Now, how this was done was, first, Griffith was trying to come up with a vaccine for pneumonia. Pneumonia was a deadly disease back then, and Griffith was working on a vaccine for pneumonia. So he selected two strains of pneumococci, and the two strains were called R strain and S strain. The R strain was called type 2R, and the S strain was type 3S. Now, why he chose these two strains was because they can infect mice. And back then, even now, mice are similar to humans in many ways, and studies in model organisms can be extrapolated to humans. With the same thing, with the same point in mind, Griffith took mice as the experimental organism, and when he infected mice with the 2R strain, it was a non-virulent strain, and the mice survived. That means this was not a harmful strain. It could not kill the mouse. The mouse survived. This is because this one does not have a protective capsule over it, does not have a protective covering over it, and this can be easily killed by the organism. Now, when he repeated the experiment with the S strain or smooth strain, which had a capsule over it to protect its core, this is the virulent strain because it could be protected from digestion by the host. The host was killed. When S strain was injected into the mouse died. So what Griffith did was, he heat killed the S strain. He exposed it to heat for so long that it died. And then the dead S strain was injected into mouse, and the mouse was alive. That means the capability of the S strain to cause death is removed when it is heated. Now, this heat-killed S strain was mixed with a live R strain, and then the experiment was performed. That is, rough live strain, smooth dead heat-killed strain were mixed together and then injected into mouse, and the mouse died. And this was surprising. And Griffith took the heart out of the dead mouse and saw that somehow the R strain, the live R strain, was converted to a smooth strain, converted to the virulent smooth strain. And he said, this smooth strain, the heat-killed smooth strain, had a transforming principle in it, which it transformed, a trait in it, which it could transform into the R strain and change it into the virulent S strain. That was Griffith's experiment. He said something transformed the R strain, and that something came from the S strain. And that he named as the transforming principle. Now, Avery McLeod and McCarthy carried this experiment forward, and they took this extract of the smooth strain of pneumococci, and they digested the DNA by using a DNase. And when they injected into mouse lived. Now what they did, they took the extract again, they digested the RNA, the protein, the fats, the polysaccharides, the carbohydrates, everything else except the DNA. In the second experiment performed by Avery McLeod and McCarthy, they digested everything except the DNA, and they injected that into mouse, and they saw that the mouse died. And through this, they confirmed that it is DNA that is the transforming principle and DNA that passes on the trait. Thank you.